Thank you very much, Eric. It definitely wasn't as bad as it could have been. I think Andy may take that later. Thank you all very much for coming, and I'd like to thank Imperial for the opportunity to come here tonight and talk about what we've been doing over the last few years. Cystic fibrosis is a disease that has changed enormously over the course of my lifetime, and as opportunities to talk about oneself don't come up very often, I thought I would seize this one shamelessly and actually make it quite a personal story that I'll talk about today. So this is me in 1963. At the time, the Beatles were top of the charts and would stay so for quite a long time, which to a Liverpool family was a very big issue. The first ever episode of Doctor Who came out 2 weeks after I was born. I don't think that the title of the episode has got anything to do with my birth. And 2 weeks before the assassination of JFK, so a time when lots was happening. I look a bit disconcerted to be propped up in a health and safety hazard. I've got no idea why I'm sitting in a laundry basket, but I'm sure my mum can explain it later. But the main thing was that I was born healthy, and had I been born at that time in the early 60s with cystic fibrosis, things really looked very grim. So you can see here cohorts of births through the years. This is the cohort that I was born in here. You can see I would have had a less than 50% chance of surviving to go to secondary school, and about a 1 in 5 chance of surviving beyond my 30th birthday. So progress was being made, but it was really pretty slow, and the reason it was pretty slow was because the disease was really rather poorly understood. It was recognized as being an inherited disease, so 2 healthy carrier parents would have a 1 in 4 chance of having a child with cystic fibrosis. But at that time it was called cystic fibrosis of the pancreas because it was largely recognized as being a condition where children failed to thrive, they malabsorbed their food, they couldn't actually get sufficient nutrition from their food. And it was a little time later that the associated chest abnormalities were identified, and it was found to be a constellation of symptoms, so a multi-system disease. Some progress was made in the 1950s when, during a heat wave, this particular paediatrician, Dorothy Anderson, noticed that multiple patients of hers were coming in with very severe dehydration and salt loss. And she put 2 and 2 together, and working with a colleague in the laboratory, Paul de Saint-Ignace, they developed a test to actually look for cystic fibrosis based on the loss of salt within the sweat. You can see here the arm of a little baby having the sweat induced, and this is still one of the gold standard tests that we use today. So there was quite a lot of progress in understanding that this seemed to be a multi-system disorder, and it was related to salt loss within the sweat. So I grew up oblivious to all of this, as Eric mentioned, in the north of England, so prior to Dundee, growing up in Blackpool, enjoying all of the things that a northern seaside town has to offer. And then progressed on to Queen Mary's School at Lytham St Anne's. And you can see here that playing field was just as bleak and freezing as it looks in the photograph. And during the ensuing few years, I was fairly exposed to medicine, having a mum who was a single-handed GP. And my sister and I would get the bus home from school and have to go and sit in the waiting room of the surgery doing our homework while the patients traipsed in and out. So it was a fairly big part of our lives. But over the next few years, I transformed myself from this little swotty, lovely angel into one of these feisty sixth formers. And it was during this time that I was thinking about doing medicine. And as part of this chemistry set here, this was the time when I really decided that I probably would go for it. And that particular maverick teacher is probably one of the reasons why I did manage to get there, because I can vividly remember him telling me not to bother because I'd never get into medical school anywhere. And that was a little bit of a challenge which I rose to. In the 1980s, I went to Dundee University Medical School, and those of you who know me well will know that I went more for the outdoor activities than for the medical school itself, but it was a fantastic education, and I did manage to qualify and graduate, and then went into paediatrics, as Eric says. But this time of my being at medical school was a really amazing time for cystic fibrosis. There were some extraordinary developments. Starting in the early 1980s, when people were just starting to piece together some of the abnormalities they were finding both in the nose and also in the sweat gland, and realizing that this was a disorder that was to do with the transfer of salts in and out of epithelial cells. And then in the mid-1980s, the gene was localized to a part of the chromosome which allowed scientists to actually hone down and identify that gene. And in 1989, the CFTR gene, as it's called, was identified. And this is has been determined to be the gene that's responsible for cystic fibrosis. There was a whole volume of the Science journal, a very, very highly esteemed journal, focused on the scientists who had actually found that gene and described where it was and what it looked like. And that's really been one of the major findings that has changed the outcome of cystic fibrosis over the last 20 years or so. Meanwhile, I was doing pediatrics and beginning to think about what I wanted to do. I had an initial dream to do infectious diseases, so I was very lucky to be trained at the Queen Elizabeth in Hackney, which sadly is now closed, and then through Great Ormond Street. And I then spent a short period of time in Africa and came back swearing never to talk again about NHS overcrowding, but that didn't last very long, as you can imagine. It was around the time I was coming back from Africa that I was looking at jobs, and there weren't any infectious disease jobs in London at the time. And I thought it would be a good idea to do some general pediatrics, maybe some respiratory and a bit of intensive care. And I found myself applying for a job at the Brompton Hospital. Now, many of you won't recognise this building because this is the building that not too long ago was sold off for a rather small price and turned into flats, I understand it. But I made an appointment to come and see someone called Dr. Andrew Bush, who sounded very serious on the phone, and I was probably a little bit daunted. Now, if you Google image Dr. Andrew Bush, you get this. You get this. And very bizarrely, you get this. So you can probably imagine it was a bit of an anticlimax when I turned up to meet Andy. And many of you have heard the story about how I came to be appointed at the Brompton. I really wasn't quite Andy's cup of tea. I probably wasn't serious enough, and I certainly wasn't academic enough. But it was just my luck that at the time that job was a mixed respiratory and cardiac job. And the cardiologists who were on the interview committee were always a little bit partial to anyone in possession of 2 X chromosomes. And I think they probably overrode Andy. To prove him right, the person that he wanted was probably a professor about 10 years ago, maybe even longer. So you were right, Andy. But anyway, I got the job, and I came to the Brompton, and I was doing cystic fibrosis for the first time and really enjoying it, and very much not just the interesting clinical disease itself, but also getting to know the families of children with a chronic disease, which has always been something I've been really interested in. So starting to think, really, was I making the right idea thinking about infectious diseases? And then Andy came up with the idea that perhaps I'd like to stay on a little bit longer and do a higher degree, and got together with this guy called Alton and suggested that I stay on and do that. Now, more fascinatingly, if you Google Alton and NHLI, and very bizarrely, I really would love to know about the melons sometime, Eric. So actually, it was rather a relief when I came along and met this unassuming chap in his laboratory with his electrical equipment. And at the time, Eric had been working in cystic fibrosis for quite some time with Duncan Geddes, who it's a delight to see here again tonight. And in fact, they had fairly recently published in Nature Genetics that really quite hot on the heels of the gene being identified, they were one of the groups who'd already managed to get the gene and put it into an animal model and demonstrate that you could actually reverse some of the hallmark features of cystic fibrosis in mice. So it was a really exciting time in Eric's lab, and it was a real privilege to be able to come and work in there. But of course, I was interested in infection. And between us all, we dreamt up this project looking at a bacterium called Pseudomonas aeruginosa. This is the main pathogen in cystic fibrosis. It's the thing that people get relatively early. It's very difficult to get rid of. And at the time, there was some literature out there saying that the way that this bacteria stuck onto the surface of the cells was different in cystic fibrosis, and that perhaps there were more receptors for those bacteria on there. So we decided to try and set up a technique to measure the way bacteria was directly sticking to cells. I had a huge amount of help at the time, not only from Andy and Eric, but also from Ty Pitt, who at the time was running the Colindale service, and from Anne Dewar, who's a complete font of knowledge in the electron microscopy service at the Brompton and has sadly since retired. But they were both fantastically helpful, and I'm really grateful for that. And we did, in fact, find, as you can see here, that if we took cells out of the nose of patients with cystic fibrosis, we would have a higher number of those bacteria sticking to them once we'd added them in the lab than in patients without cystic fibrosis. There's about 14 patients here, mostly women, because the men largely ran away screaming when they saw me coming towards them with their brush, and particularly Andy, from what I recall. The next question that we said was, could we correct those cells by actually getting the gene and putting it into the cells in the laboratory? And it was very exciting to see that we could. So these are the same cells from the same people treated with either a dummy gene or with the CFTR gene, and you can see that levels of those bacteria come down. And as a first paper in a research career, this was really very, very exciting, and actually made the front of the Red Journal, which was probably a bit of a highlight at the time. I ended up staying on for a year longer than I'd planned, because Eric and Duncan at the time were planning to do a clinical trial actually giving the gene to patients with cystic fibrosis. And so I stayed on to actually apply this in the context of the clinical trial, which was really fun and interesting. But I did have to finish clinical training, and as Eric said, I went back to Great Ormond Street, and it was great to be trained by Bob Dinwiddie and Colin Wallace. But I also spent a year working with the infectious disease team there, Nigel Klein and Vas Novelli. And that was the start of a collaboration that went on over the next few years, because Nigel had a particular interest in this compound called mannose binding lectin. So what this compound is, it's something that is produced by the body and is a host defense protein. So it actually binds to bacteria and allows our bodies to rid ourselves of the bacteria. And there was interest in the time at looking to see whether or not this might be what we call a modifier gene of cystic fibrosis. So if you had more of this protein, you might actually keep your lungs healthier with CF. So we did some experiments to look at whether or not we could actually see that this protein bound to these bacteria. And you'll recall this is the one, Pseudomonas, that I was very interested in. Disappointingly, there was almost no binding. But in contrast, there was a huge amount of binding to this really nasty organism called Burkholderia cepacia, which at the time was just coming out into epidemics, and a number of CF patients were actually dying from it. So it was quite interesting to find that, and that the amount of binding actually correlated with the amount of activation of the inflammatory system, so that could be really quite damaging to patients. So 1999 came around. It was quite a big year, the year that saw the launch of the Euro. My favourite author, Iris Murdoch, died, and the Scottish Parliament got set up. But most importantly of all, I came back, as Eric mentioned, as a senior lecturer. It was a real dream job for me, not just because of the science and the clinical, but actually to come back and work with all the people that I'd really enjoyed working with before. It's really fantastic, and it has borne out to be as good as I'd hoped. So I'm going to talk for the rest of the lecture about what we've been doing in the ensuing 10, 15 years. But I thought it was probably for those people who aren't directly involved in cystic fibrosis, and also for those people who perhaps aren't medical at all, it would be useful to just sort of say, where are we with cystic fibrosis now? What do we actually know about it? Because an awful lot has happened. So this is what we know the protein looks like that is made by the CF gene in normal people who don't have cystic fibrosis. And this thing here is actually the surface of the cell. And the protein, the CFTR protein, sits in the surface of the cell, and it forms a hole or a pore. And this part of the protein can move in or out of that pore, a little bit like a plug moves in and out of a sink, and it can cause that pore to be open or closed. And there are a number of functions of this protein, but the main one that we associate with disease is related to the passage of salts in and out of the cell. So you can see here that when the channel is open, chloride salt can go out onto the cell surface, and it also speaks to one of its neighbouring proteins, which is involved in sodium transport. And by doing that, it actually regulates the amount of those salts on the cell surface and regulates the amount of water that stays on the cell surface. And in the lungs, this is particularly important. So just to sort of talk you through this, this is a diagram of a normal lung cell. Here's the airway here. This is a normal cell with the normal CFTR gene, and it produces this protein that allows chloride out. It talks to its neighbouring protein, and it allows a small amount of sodium in. And what we see instead in cystic fibrosis is either the protein's not made or it's in the wrong part of the cell. The chloride can't get out, but too much sodium gets dragged in because this talking has failed somehow. And as you'll remember from A-level chemistry, when sodium goes somewhere, water follows it. So the surface of the cell effectively gets very dehydrated. And these have been nicely shown in experiments from Chapel Hill, which really has led the field in understanding this basic mechanism. So these are Petri dishes of cells here with a fixed amount of a green fluorescent liquid put on top at the start of the experiment. These are the normal cells, and these are the CF cells. And then they go home and come back 24 hours later. And I think you can see what's happened to these cell cultures. the normal ones look pretty similar to they did before, and the cystic fibrosis ones have drunk up that fluid. So they've now got only a very small amount of liquid on the surface, and that's important for two reasons. And the first of those is shown here. This is the surface of the cell, and on the surface of airway cells there's these little hairs that stick up called cilia, and the cilia beat constantly as a defense mechanism to rid the body of any inhaled pollutants or bacteria or viruses. And these hairs can only actually beat when they're able to stand up because they've got enough liquid around them. And so what happens in cystic fibrosis is there's too little liquid, the cilia are all bent over, and they don't actually move properly. Possibly also important, though, you can see these little red blobs which have been put on here. They're supposed to illustrate inhaled bacteria or viruses. And in this slide, the little red blobs aren't actually anywhere near the cell surface, but in this slide, they're getting actually quite close. And when they get close, they make that cell become inflamed. And inflammation is one of the really big hallmarks of cystic fibrosis and one of the biggest problems. So the reason this is so important is our entire airway is lined by these cells with these cilia projecting from them. And the first line of defense is for them to beat and clear the airway of anything that lands on it. So in cystic fibrosis, that doesn't happen. So this is the sort of cascade through from the gene mutations to abnormal functioning of this protein, depletion of the airway surface liquid, the mucociliary clearance, which is how those cilia beat, fails, and we then enter a vicious cycle of bacteria coming into the airway becoming very inflamed, the cells getting damaged, and then that being a breeding ground for more bacteria. And eventually lung scarring happens, as you can see in this CT here, and end-stage lung disease. And very sadly, even now, the majority of patients with cystic fibrosis die of respiratory failure. And when they get to that stage, the only possible hope is a lung transplant. One thing that has really changed the field quite dramatically over the last few years or so is that newborn screening is now done for cystic fibrosis. So when babies are born, within the first few days of life, they have a little blood spot taken. And previously, we had to wait for children to actually develop the signs of the illness and actually get quite unwell before they were tested. But you can see now that the vast majority of babies are diagnosed within the first month or so of life, so giving us the opportunity to intervene very early and hopefully to slow the progression of the lung disease. Nevertheless, when we do diagnose it, we ask those families and those children to embark upon a very, very rigorous regime of treatment. We have to ask them to do physiotherapy constantly to clear the airways of mucus and infection. We give them a number of inhaled medications to help with that. We get them on a lot of antibiotics. They often have to come into hospital and have intravenous antibiotics. We use a large number of other drugs. And you can see just some of those here. So this is a very, very big deal. And for patients with all but the very mildest disease, this can take several hours a day. The drugs can have side effects. And what we're doing currently in the clinic is treating the symptoms of the disease rather than the cause itself. And you can see here some rather better survival figures, the median predicted survival between these years here. So if we look at the top of these in 5-year bands, the median predictive survival is still only in the mid to late 30s. And I thought many of you will realize, despite all of this input, which impacts the quality of life, most of us really just don't think that's good enough. And that's the reason why people are researching this field so vigorously. So the areas that we've been researching, as Eric was mentioning, we're trying to understand the early disease processes, taking the opportunity of looking at young children with cystic fibrosis, particularly with regards to infection and the inflammation that happens, and then the remodeling, which is the early stages of scarring within the airways. We're seeing if we can get better ways of detecting early lung disease and infection. And ultimately, the aim, of course, is to see if we can get better treatments. So I'll just spend some time going through a couple of those and tell you what the groups that I'm working with have been doing. With regards to the first of those, understanding the mechanisms of disease, we're very fortunate here that we have an active research program using bronchoscopic samples from patients with cystic fibrosis. So nobody gets put to sleep purely for this, but when a child is having a bronchoscopy anyway, we take the opportunity of getting samples. You can see here, this is a flexible telescope that goes down a breathing tube into the lungs of a patient with CF. You can see here actually what we're looking at, and it gives us the opportunity to take very small pinhead-sized biopsies of the airway, and then to section and stain those and look for all sorts of changes. We also take washings from the airway, and we can look for inflammatory cells and inflammatory proteins in that. Everyone always says, oh dear, but is that safe? We've done an awful lot of work to show that adding this onto a bronchoscopy, which is otherwise needed, is completely safe, and actually adds very little in the way of time once we get used to that. So there's a number of things that we've been able to do with these techniques, and one of them is to look actually at the basic mechanism of disease, so look at the transport of those salts across the cells. And that's important because if we're looking for ways to correct the disease, we need to have something to measure in the end. So you'll recognize this from previously. This is just to show you that, or to remind you that when positively and negatively charged ions pass across a surface, that sets up a system which is a little bit like a mini battery. That's how batteries work. And so we can measure the ion channel function with a little voltmeter. And this is a technique which Eric and Duncan set up called nasal potential difference. So there's a little catheter going into this volunteer's nose here, and it's connected via an electrode to a voltmeter, and we can read it out on the computer. And we can challenge the surface of the nose to respond to different types of stimuli. And what maybe you could just focus on for ease is the difference between this part of this trace and this part of this trace. So at this point, we've challenged this nose to secrete chloride. We've given it an environment in which normally, if it could, it would secrete chloride, and this healthy person is able to do that, and this CF person is not able to do it at all. Noses are useful because they're easy to get to. You can get to them lots of times, and you can dose them, and you can measure things, but actually they're not where the trouble is. And so one of the things that we took on when I came here was to develop a technique that Eric had been working on and actually measure this down in the lower airway. So when we were doing an intervention, we could actually look at the changes down there. So this is an image down a bronchoscope of a catheter that we designed to go right down the bronchoscope to perfuse some of those solutions onto the airway and to measure the changes. And when Donna McShane was here as our research fellow, the first of several, we were able to do this in quite a large number of children with cystic fibrosis and without. And similar to the diagram here, you can see that when these airways are challenged to secrete chloride, the CF ones absolutely can't, and the non-CF ones can to a reasonable extent. And the fact that they separate out quite well gives us a nice opportunity for trying to measure the outcome to an intervention. And this sort of measurement has actually now gone into 2 of our gene therapy trials. We're actually measuring down the bronchoscope, does the gene therapy make the channel, and is that channel working? We've been very interested in host defense and inflammation, and leading on from some of the early work we did with Nigel Klein, Katie Fiddler was appointed as a shared PhD student, and she took the mannose binding lectin story a little bit further. We found that mannose binding lectin, in fact, was a modifier gene for patients, so adult patients who had low levels of this protein had worse lung disease with cystic fibrosis than if they didn't, leading, of course, to the possibility that at some point a replacement treatment might be viable. Katie was the first person to report that this protein was actually secreted into the inflamed airway, so therefore it could be acting as a host defense protein. And she then went on to do some further interesting studies looking at the importance of the protein in very sick children on intensive care, and the group has taken that further, looking at the importance of the protein in neutropenic children. So children who are having chemotherapy for cancer, this protein is particularly important, and that might be one of the areas where replacement therapy is actually going to lead to success. Nicholas Regami came over on an ERS travel fellowship from Switzerland and worked with us for a few years, and he did a number of these studies, and one of them was to question whether or not, if we just take washings from the airway surface, which is what the majority of people do, are we actually getting a proper reflection of the inflammatory changes that are going on in the airway wall? Because it's the airway wall and the destruction of that that leads to the death of the patients, and yet most of us are just quite happy to wash the surface. And he found that no, in fact, they don't reflect it at all. So this is a washing, and you can see these little bitty cells here are neutrophils. And in cystic fibrosis patients, the vast majority of inflammatory cells are neutrophils. But if we actually take a biopsy of the airway wall, it's very rare to see neutrophils. Most of what we're seeing are lymphocytes. And that means that the inflammation that's going on at that level, and which might be causing the airway destruction, is of a completely different cellular nature. Now, Hui Leng Tan and Claire Lloyd, when they were working with us, actually took this a little bit further, and at the time there was a new class of cell which had been identified, Th17. We're used to thinking of Th1 and Th2, but Th17 was identified, and Hui Leng spent a lot of time setting up a stain for this and was able to actually develop an antibody stain that worked in the biopsies of these patients. And then looked at the number of cells in the biopsies of patients with cystic fibrosis and with other diseases. And interesting, you can see here, these are the controls. These are patients who don't have airway disease. They've been having a bronchoscopy for some other reason, or we've found that it's completely normal. These are established CF patients, so they've had their CF for a while. But rather worryingly, these are our newborns, so new diagnosis of cystic fibrosis. Really early in life, we're already seeing that there's an increased number of those inflammatory cells, which make it even more important that we're able to make this diagnosis early and think about acting. Now, I haven't got time to go through all the projects, but it's been a really fertile area of research, and I'm grateful to all the research fellows who've helped with it. Sarah Brown's done a lot of work and is just writing up her MD at the moment, looking at resolution of inflammation and whether that's abnormal in cystic fibrosis. And Becky Thursfield has left now to go to a consultant job in Alder Hey, and unfortunately can't be here, but she's done nice work looking at inflammation in newborn screened babies, and also looking at the role of vitamin D, because I think for a while people were thinking that was particularly important in airways disease. We've had a look at remodeling as well, as I said, the very early stages before scarring happens, and Tom Hilliard did some nice work looking in the lavages, so in the washings of these patients, and found very, very low levels in controls of these breakdown products, but very high levels in CF patients. So the tissue of the airways is actually getting broken down by some of the inflammation that's going on. It's directly correlated with inflammatory cells. And it also is inversely correlated with lung function. So once this starts to happen, the airways are getting damaged and the lung function of the patient becomes impaired. And he went on to make some further measurements looking at what we call basement membrane thickening. You can see here this is just underneath the surface epithelium, and this pale pink area here, which is rather thickened, and which is certainly abnormally thickened in cystic fibrosis patients. I don't think we've completely understand this, and we may need to think about this a bit further. It didn't seem to be related to inflammation, and it's as if there are two parallel processes going on, one of them inflammation and the other one perhaps remodeling, and I think we need to probably work a bit harder to try and understand that. And then another thing that Nicholas did was actually to spend a lot of time counting the smooth muscle cells in the airway and demonstrating that cystic fibrosis, similar to asthma, although not quite to the same extent, has an increased amount of smooth muscle in the airway, and that it may be hyperactivity of that smooth muscle which leads to some of the asthmatic-like components in cystic fibrosis. So I think we're really, really pleased at how this series of projects has gone. We've built up over the last 8 years or so quite a nice biobank of samples from patients. One of the nice spin-offs from these sort of occasions is that there might be somebody sitting in the audience who thinks, oh, I'd like some of those samples, and we could get together on a collaborative project. So please do get in touch if you think that there would be something that would be worth studying, because it's a very rich resource. So I'm going to come on to the bugs part of this lecture now and talk about Pseudomonas. As I said, it is the most prevalent infection in patients with CF. You can see here from this graph that it's not so common in early childhood, but as patients get older, it becomes by far the dominant organism. And most patients will eventually acquire this organism, and once they get it, it's very difficult to get rid of, and it becomes chronic. Treatment's very effective if we pick it up early and we treat it aggressively early. But once it becomes chronic, it's very, very difficult to treat. And one of the reasons for that is shown here. So rather than just isolated bacteria, these bacteria clump together and live in a sort of colony in a thing called a biofilm. And the biofilm actually protects the bacteria from the host defenses and also from the bacteria that we give. So we try hard to treat with antibiotics, and the side effects of those antibiotics build up as patients get older. We know if we look at cohorts of adult patients that there's increased rates of hearing problems and also kidney problems, and yet still we don't manage to get rid of the bacteria. So it is a very real problem, and it is associated with worse outcomes. So there's a number of projects ongoing in this field at the moment that I'll tell you about. One is, can we pick it up better? We know that the majority of children, when they come to clinic, can't be told just to cough up a sample of phlegm into a pot. They haven't got the sort of ability to do that, they're not productive enough. And in fact, many of our adults who are keeping well are unable to do that as well. So in order to treat the infections, we do need to identify them, but we're relying on very sort of insensitive tools. Cough swabs, where we just get them to cough onto a swab and we try and culture that, or bronchoscopy, where we go down the bronchoscope, and that's quite a good way of getting bugs out, but we can't keep doing that to children or adults, obviously. So the question that we're answering in this project, which is being done by Rishi Pabari, who's just coming to the end of his MD, is do the bacteria produce volatile gases that we can actually pick up if we just got people to blow into a bag of gas? And the answer is yes, in part. So these are some of Rishi's data. And you can see here CF patients with Pseudomonas and without Pseudomonas. There are a number of compounds, I won't go into what the chemical structures are, that are raised in patients with Pseudomonas. And then interestingly, there's this one here which is actually lower in patients with Pseudomonas. And if you put all of these data into an analysis program which generates a sort of sensitivity specificity diagram called a ROC curve, you can see here that this isn't as good as it could be. What we're really looking for is a line which goes much more up here. But beginning to get an idea that there are volatiles which are produced by these organisms, which, if we were clever enough, we could probably tease out from a breath sample whether or not somebody had Pseudomonas. This is an area that we're taking forward and that I'll talk about in a moment. In collaboration with Hugh Williams, we've been looking for a while at some of the noxious products that Pseudomonas produces, and one of those that we've identified is cyanide. We took sputum samples from patients with and without Pseudomonas, and then we developed a sensitive assay to actually measure the cyanide in those. And this is the group who didn't have Pseudomonas, and then this is a group who did have Pseudomonas. And you can see that the majority of those with Pseudomonas have some cyanide detectable, and it's not detectable at all in the others. And it was very interesting to see, actually, that that appeared to have an impact on lung function. So patients who did have cyanide present in their sputum had worse lung function than those who didn't. So an interesting finding which we've been trying to figure out a little bit more. And one of Hugh's other students, Chandrika Nair, has taken this forward and has actually looked at the effect of cyanide on the cells themselves. So you can see here, this is a cell. Here are the cilia. These cilia are beating constantly, and we can measure that by certain tools down the microscope. And what she's shown is that, unlike the control cells which carry on beating if we leave them in culture for a while, if we put cyanide on the surface of those cells, the cilia just start to beat very slowly. And so it may be that the bacteria are getting into the airway, and then once they're there, they're clever enough to produce chemicals which enable themselves to stay rather than getting cleared. So what about treatments? I've mentioned some of the limitations to antibiotics. And although new antibiotics are being developed, they're really not being developed very quickly. It's rather more taking old antibiotics and putting them into new forms of inhalers, etc. So we and others are starting to think of alternative ways of doing that. And there's one quite attractive possibility, and that's the use of bacteriophage. What bacteriophage are viruses that are specific for bacteria. And you can see one here. They latch onto the surface of bacteria, and then they've got a tail through which they inject themselves into the cytoplasm of the bacteria and cause bacterial destruction. And bacteriophage have been around a very long time, long before the invention of antibiotics. They were available, but they've largely been developed in some Eastern European countries and in Russia, and they haven't undergone the rigorous testing that most of the Western world would expect, and so therefore they've, I think, probably largely been somewhat dismissed until recently, when people are becoming a bit more interested in them. So as the other part of Rishi's project, what he's doing is he's taking bacteriophage specific for Pseudomonas, infecting mice with Pseudomonas, and then giving them the bacteriophage either at the same time or a little bit later, or even preventatively. And the data are very, very encouraging. This is a log scale, which means that these levels are extremely high. So when we infect these mice with bacteria and we then sacrifice them 24 hours later, they've got masses and masses of bacteria in their lungs and actually are often looking very unwell. These are the mice which are given the bacteriophage as well. So with the exception of one mouse here, every single other one has killed all of those bacteria with the co-administration of bacteriophage. And nicely backing that up, no matter which inflammatory cells you look at, you see a significant impact on inflammation. So these phage are capable of killing the bacteria and also of reducing the inflammation, which is a particular problem in CF. So all of this focus on infection has led to a recent grant that I'm really, really excited that we've been given. The Cystic Fibrosis Trust has set up a series of new strategic research centres, and we put in a bid on one, bringing together all of this infection work, looking at Pseudomonas infection, better detection, understanding, and better treatment. And we'll be opening this centre probably in about November. But this brings together a really fantastic group of collaborators, both from within Imperial and also outside. There's some European collaborators, and we've also got help from a small biotech company who are making the bacteriophage. And I'm really, really pleased to have Ty Pitt back on board. Thank you so much, Ty, for agreeing to take part and looking at the oversight of this centre. So what will happen here is we will get 5 PhD students for a period of 3 years, and they will work in parallel to try and take some of these findings forward to actually turn into a reality the breath test, if it's ever going to work, and do a clinical trial of the bacteriophage and see whether we can get that to work. So it's a very, very exciting time, and I'm really pleased to be doing this. So for the last 10 minutes or so, I'm going to talk about clinical trials of new treatments. And the new treatments that we're particularly interested in are those that target the basic defect. You'll have heard me say that the problem with the treatments we're using in the clinic is they're all treating the symptoms, they're not actually treating the root cause of the disease. So most people think that if we could successfully target the root cause of the disease, we'd have a lot more chance of serious success. So there are 2 ways of doing that, either targeting the gene itself or targeting the protein. I'll talk first about replacing the gene so that the gene can make the new protein. So what we mean by gene therapy then is trying to make a cystic fibrosis airway cell into a healthy cell. You can see here these are normal cells. That blue thing is the nucleus, and the nucleus has the right machinery in it to produce this orange protein, which is the CFTR protein sitting in the right place at the cell surface. These are the CF cells which don't have the right machinery to do that, and that protein therefore isn't there. So what we're trying to do is to take a normal copy of the CF gene, get it into the cell, into the nucleus, and make that nucleus actually produce the protein. And that's the basis of gene therapy, at least for cystic fibrosis. So you'll recall that I said the gene was discovered in 1989. It was quite quickly worked out what it was doing, and then very, very quickly experiments in the laboratory showed that you could take a normal copy of the gene and turn a CF cell into a non-CF cell. And this was around the time that Eric was doing the animal studies. Very quickly they went into clinical trials in the nose of patients with CF, and then actually quite quickly also into the lung. But what's happened since that time is there's been a lot of reiterating back and forth to try and actually make this system work even better. To summarize, there's been over 20 clinical trials. Almost all of them have used a single dose, usually given into the nose because it's so easy to measure. And then the measurements that people have made have been things like the potential difference that I showed you earlier. Can we actually measure something on the millivoltage, or taking cells out and saying, can we see that the gene is in there? The things that have been learned, I think, about that is that it's very difficult to get that DNA carried into the cell by a virus if you want to do it more than once. There are other ways to do it, but viruses, it's difficult to do more than once. And there's only been one program to date that's actually looked for clinical benefit. Most of them are looking for evidence that the gene is in the cell, but looking for clinical benefit, there was one program, and unfortunately that didn't work. So it seemed that we got to the stage where we had proof that we could get the gene into the cells, but we didn't know really what the clinical relevance was. So these are some data from the trial I mentioned that Eric was doing when I was coming up to the end of my time in the lab. And this is making those electrical measurements down on the airway surface. And this axis here is a measure of chloride secretion. These are patients given a placebo, and these are patients treated with the active gene, single dose. Before and after placebo, so they couldn't secrete chloride, and then before and after the gene therapy, we saw a significant improvement in the amount of chloride that they could secrete, probably to around 25% of the normal levels we look about here. So that was really exciting. It was the first time that the basic defect had been changed in the lower airway, but there were some problems with that. It was quite short-lived. These measurements were made quite soon after that dose. And also the patients who had the active treatment got a little bit of a flu-like illness. They tolerated it well and they just took some paracetamol, but it wouldn't be the sort of thing that you'd be wanting to give patients with cystic fibrosis repeatedly. We didn't really know what any of these results meant, and certainly we didn't know how much gene transfer had got into the cells, or even how much cells were needed to be corrected to see what in terms of a clinical outcome. And it was to try and grapple with some of these questions that the UKCF Gene Therapy Consortium was established, coming up for 10 years ago now. So at that time, there were 3 centres in the UK who were doing gene therapy for cystic fibrosis. It was Eric's group at Brompton Imperial, Oxford and Edinburgh. And they were brought together by funding from the Cystic Fibrosis Trust to form a consortium. So within that consortium, they were working together in a streamlined fashion without competition and sharing core facilities. And the goal that was set at the beginning of this time was to take the best currently available vector, so the thing that gets the gene into the cell, and put it into a trial which was designed different from all those others. So designed in a way that we could say, does it make someone better? So looking at clinical outcomes. The 2 main questions were, which would that best product be, and how would we know success when we saw it? I won't go into this in any detail, but the consortium clinical program has been huge. It's taken around 70 people full-time working for the last sort of 7 or 8 years to try and address these questions. But where we've got to so far is that we've found a formulation that is repeatable. We showed fairly early on that we could get fantastic gene expression. This is a gene that turns cells blue, and you can get fantastic gene expression with a virus, but you just get it once. When you try and give that virus again, you get a sort of vaccine response, and the virus doesn't get in. So we knew fairly early on that at least at this stage we had to be using something that wasn't a virus. We had to be able to get it in through a nebulizer. Some of the early trials actually put things down a bronchoscope, but we couldn't do that repeatedly to people. And we had to be able to manufacture it according to the current standards of drugs that we give to people in trials. It had to have a good toxicity profile. And we tested a very, very large number of different products to try and get them to meet all of these criteria. And in the end, just came up with 3, which would have been useful. And those went through some preclinical testing, and we came up with this vector here, which is a liposome. So it's a synthetic vector, not a virus, which we can couple with the DNA and get it into the cells. I mentioned that the last trial showed really quite short-lived expression, and this was the, one of the components of that vector that we used in that time. So we got a lovely peak here, but it tailed off, and by about a week it was gone. And our reasoning was that if we're going to do this, we can do it repeatedly, but we don't want to be doing it every couple of days. It's going to be very cumbersome for the patients. So one of the major improvements that was made by the Oxford group of our collaborators was to change the makeup of the DNA. And what they've got now is a slower switch of the gene on, but when it switches on, it stays on for much longer. And in fact, that graph goes out to 2 months now, so we know that we can get gene expression for a much longer period of time. And this has influenced our choice in the current clinical trial of dosing interval. So this is what our clinical product looks like. Here's the lipid. We have to reconstitute it on the day. Here's the 169th bit of circular DNA that was created by the Oxford part of the consortium to test and prove that this was the optimal one. And this is the one that's got that long duration and several other features that make it very attractive. We did a single-dose trial of this combination because although gene therapy has been into patients before, we'd never actually done this combination, and we had to show that it was safe. And Gwyneth Davies, when she was here, based her PhD around this. We gave nebulized doses to patients, and we had a look at the outcomes. And we found a dose which was safe. The higher doses gave a slight flu-like effect, but at this 5 ml dose in a nebulizer, it didn't. And we also saw some quite encouraging results. We weren't intending particularly to see these results after a single dose, but when we measured the nasal electrical changes, some patients had really long-lasting and quite dramatic changes. When we measured in the lower airway, we were already starting to see some of those changes. And in fact, to our surprise, we began to see some physiological effects as well. So a sensitive measure of lung function, the lung clearance index, looked as though that might actually be improving in even patients who'd only had 1 dose. So on the back of that, we went into a multi-dose clinical trial, giving the gene therapy repeatedly over a course of a year, every month, reckoning that that was probably the time period after which we might expect to see some clinical benefit. We're looking at measures of lung function. The primary outcome is a measure called FEV1. We've got a lot of other outcomes as well. And these patients are dosed in cubicles within our respiratory biomedical research unit, both here and at the Edinburgh site. I don't think the research team usually looks quite as glamorous as this. This must have been posed for the photograph, I think. But they get a nebulizer inside this cubicle so that the gene therapy is not coming out and going into the environment. And then they undergo a massive number of investigations. And what we're trying to do, although we're looking for lung function to improve, we're also trying to anchor that to a large number of other investigations, CT scans, blood tests, bronchoscopies in a subgroup of them. I'm really delighted that we've actually finished dosing now. This has been a very, very long time coming, and the trial has been an enormous effort from everyone. 116 patients across the 2 sites have completed dosing. We're still doing a few of the follow-up visits to see how they've responded, and then we need to process all the samples and get the data entered and analysed. But by the end of summer or the beginning of autumn, we should have the results, and we will know whether this trial has actually failed or succeeded. We'll be presenting the results at the North American CF conference in October, and of course we're very, very hopeful that it's going to have worked, but we're not unrealistic. There is a possibility that it won't have done. And in order to sort of future-proof for that possibility, in parallel to this Wave 1 program, the Consortium's been working very hard on a Wave 2 product. And this Wave 2 product, we think, is probably the first virus that can be given more than once and can lead to very long-lasting expression. This has been created between the scientists, both at Imperial and at Oxford in particular, and is a modified lentivirus for which the Consortium won a couple of awards a couple of years ago. So we're working very hard on getting that through in parallel, so that if this doesn't work, or if it doesn't work as much as we need it to, we will have a plan B to go on to. So finally, just a couple of slides on coming one step down and trying to make the protein work. There's been some fantastic progress in this over the last few years, which has been really exciting to be involved with at the clinical trial stage. Ivacaftor is a small molecule drug which can be taken by mouth and which activates a certain type of cystic fibrosis mutation. This is a G551D mutation. It's not the commonest one. It's actually quite a rare mutation. But with this mutation, the protein gets to the cell surface, but it's jammed shut, a bit like the plug is jammed in the plug hole. So the salts can't get out onto the cell surface. And what this drug does is it binds to the protein, allows it to open, and allows it to function much more normally. And at the Brompton, we were part of 2 of the phase 3 trials, which we gave this drug to both adults and children with cystic fibrosis. And just a few of the results. This is change in lung function. There was a 10% improvement in lung function within the first 2 weeks of starting the drug, and that improvement was then sustained for the rest of the year of the trial. There was a significant improvement in the number of times those patients had exacerbations and got admitted to hospital, and there was a significant improvement in weight gain. And these results were very similar in the adults and in the children. They led to the drug being licensed, and we've been able to prescribe it to our patients for the last year or so, but it is a very small minority of patients. We're very pleased that the Brompton's leading on a global trial of this drug in preschool children, so 2 to 5-year-olds, who are actually getting this drug at the moment, and the results are being analysed to see whether or not that's safe in that age group. Importantly, we need to try and expand this sort of approach into other types of mutations as well, because it's about 4 or 5% of patients who currently are getting this. There have been some other trials which show promise in other very, very rare mutations, but the common mutations, which is where probably most people are trying to go, despite a lot of trial activity, it's currently looking as though that's going to be very, very much harder. It might be much more difficult with these oral drugs to correct the common mutations, and it may be that gene therapy is actually going to do a better job for those patients. The reason we've been able to do so much of this trial activity is that we, a couple of years ago, became one of the sites for the European CF Society Clinical Trials Network, which is a network of different sites across Europe, all trying to standardize and streamline their procedures and make sure that they are conducting trials in a very, very standardized way. I'm doing this in collaboration with Dr. Diana Bilton from the Adult Service, who's the co-PI, but also just want to acknowledge the input from the adult physicians who've been fantastic in helping with recruiting these patients, and particularly to acknowledge Sandra Scott. I don't have a photo of her, but she does make exceedingly good cakes. She is a very experienced adult CF trials coordinator, and she has been fantastically helpful, both in herself but also in training others around her to actually get these trials working. And the other benefit of the network is that fairly recently we've been appointed running a core facility for this sensitive outcome measure of lung clearance index. So Claire Saunders, which you can see on the right here is one of our physiologists who's going to be running that facility with the help of Katie Bayfield and others within our group. And we're really pleased to be able to provide that for the benefit of the European community. I think I'm coming to the end, but I think probably in terms of looking forward, one of the themes of this talk is that I think a lot of the successes have come out of big groups working together, either the CF Gene Therapy Consortium or the European Clinical Trials Network. And I think that that probably is the way that the future's going, is no matter how intelligent and how hard-working we are, we're not going to achieve success by beavering away in isolation. And I think this is probably one of the lessons, at least of the last 10 years, has come forward. So I just want to say a few thank yous. Firstly to my family, to my husband Neil, who has been a most amazing support to me and a fantastic dad to our 2 children, Oscar and Scarlett, to my sister Julie, and also to my mum Val. You can see her here. This is her at the age of 75 doing her first skydive. So she's going to be a bit of a hard act to follow in terms of energy levels, but I will try my best. Also to thank friends and old friends and newer friends who have been really fantastic in making the downtime so good. These are those feisty teenagers that you saw in the Queen Mary School common room. Some of them are even feistier now than they were then. And also newer friends. I think I haven't got time to thank everybody in terms of collaborators, colleagues, fellows, clinical teams, etc. But let me just sort of say again that I do think I've got a most fantastic job, and 99% of my enjoyment comes from working with the people in this room and on these slides. It's a real pleasure, and I'll probably be here forever because I can't imagine another group of people with whom it would be such fun to work. But finally, I do really want to focus on Andy and Eric. As my kids would say, they are awesome. They have been fantastically supportive of me throughout my career. They've given me opportunities, they've picked me up and dusted me down when those opportunities haven't gone so well, and they've been fantastic celebrating when things have gone well. So I just really want to thank you both very much. I definitely would not be here if it weren't for you, and it's a great, great pleasure to be here and to carry on working together. And I fear that Andy might want to say a few words. Thank you very much for listening.